Hello, today we're moving on to our second of three microorganisms. So far we have studied yeast, which we know has never appeared on a long question on our home economics exam. However, bacteria has appeared on two occasions, and the first of those occasions was in 2013 on question 3 on section B. Then we had to wait 6 more years, and in 2019, again on question 3 on section B, bacteria appeared again. Now, have you noticed there that that was question 3 on both years? Now, it's not set in stone, but we all know that question 3 is always going to be related to food studies area, and it tends to be on preservation, whether it be domestic or commercial preservation, also on food processing, packaging, labelling, things like HACCP, sensory analysis, and of course the microorganisms. That tends to be what we see on question 3B, but like I say, that is not 100% guaranteed. Now, our third microorganism is mould, which we'll be looking at in a later chapter. So today we're just going to look at classification. Guess what? We're back to that word again for about the 150th time. And as you know, when we classify, we divide into groups. Now, any of you that are doing microbiology in your biology course will know that most of the time we classify bacteria, or we group them according to their shape. If you are asked on a question to classify bacteria, always go with that as being the main classification. However, in home economics, we have 3 more different ways of classifying bacteria other than shape, which we're going to look at today. So bacteria, what do we know about them? They are unicellular. Now, we had that word already on our chapter on yeast. Unicellular means that we have one single cell. Remember, yeast was this oval-shaped, single-celled fungi. We find bacteria in air, soil, and water. For example, you are probably aware that bacteria can travel in the air, but also soil. For example, the bacteria Salmonella can live in soil, and we have to be very cautious and careful about uncooked cookie dough. Now, you're all aware that we can buy uncooked cookie dough in the supermarket. People can slice it up in discs, bake it in the oven, but some people actually like to eat raw cookie dough. For example, tubs of ice cream, you often have raw cookie dough. But wheat obviously will grow in soil. That wheat is used to make flour. The flour is used to make the cookie dough. So we need to be particularly careful about our source of cookie dough. And certainly if you are purchasing raw cookie dough in the supermarket, be cautious with regard to the use by date. Plus, we find bacteria in water. For example, there's a very, very dangerous bacteria called Legionella, and it can bring about what's called Legionnaire's disease, which can be particularly harmful. It can actually kill somebody if, for example, they had an open cut or open wound and the Legionella was to get into their cut or open wound. And Legionella actually like a very warm, moist environment, and not being well, probably I'm being a bit extreme here, but it's the one thing I do have to say whenever I'd go to a hotel on holidays, etc., always run the shower because around the shower head, you know, that will be the perfect warm, moist environment for Legionella. But then again, I can be a bit extreme. Moving on there. They are also found on plants and in animals. We know, for example, we talked about kimchi, a fermented food. And we said they how to make kimchi, which is a Korean dish where they shred up all the different vegetables like Chinese leaves, carrots, etc. And bacteria on the surface of the vegetables in an anaerobic environment, i.e. locked in an airtight jar, will start to feed off the natural sugars in the chopped vegetables, producing lactic acid, and we end up with this fermented food, kimchi. In humans, we find bacteria in our nose, our mouth, our throat, on the surface of our skin, and we have mentioned quite a few times now about the fact that there are trillions of bacteria living in our large intestine or our colon. And a term that you might have heard being tossed around in general conversation is the microbiome. And the microbiome is just a collective term for all of the bacteria that make up one human. And we are told to look after our microbiome and to note that most of our bacteria in our body live in our bowel or our large intestine. Now I'm going to highlight the next one because it's very, very important. And that is, we said when we were looking at the chapter on milk, that milk will be, I suppose, a magnet for bacteria, for want of a better description. Bacteria absolutely love moist food. Anywhere there's moisture and a source of nutrition, bacteria will be there. For example, milk, stock like chicken stock, beef stock, raw meat, raw fish, all high in moisture content. So we need to be particularly careful when we're storing these foods because of the fact that they will attract bacteria so much. We also should know, and I'm going to highlight that as well, that bacteria have beneficial and harmful effects. In other words, they have positive role and they have a negative role. Just to give one example for each, a beneficial role of bacteria is that it's used in stage 2 of vinegar production. Remember, acetobacter added to cider, to red wine, and we get our vinegar. We also will have bacteria like, remember, Lactobacillus helveticus in the production of cheese, converting lactose to lactic acid. And when we move on to another chapter called milk products, we'll talk about Lactobacillus bulgaricus used in the production of yogurt. In terms of harmful effects, as this is a food-based course, of course bacteria can bring about the spoilage of food. We saw that in the natural souring of milk, where naturally occurring lactobacillus bacteria, not killed during pasteurization, over a number of days in a fridge can produce enough lactic acid to cause the milk to sour or to spoil. So bacteria do not have any chlorophyll. Now we know from our study of carbohydrates, in order for food to be made and photosynthesis to take place, chlorophyll needs to be present. However, humans, bacteria do not have chlorophyll, therefore cannot make their own food, so the food has to come from an outside source. Now that outside source can either be what we call parasitic or saprophytic. Parasites are bacteria that live off living matter. Saprophytes live off dead or decaying matter. Again, this is a food-based course, so we will talk about saprophytic bacteria. So on our home economics course, we study saprophytic bacteria that secrete enzymes into dead or decaying matter. Now that will lead us on to mode of nutrition of bacteria. And what I would say to you is see notes. Now you might decide to put this somewhere towards the end of your chapter on bacteria, but hopefully you have a hardback copy where you're taking extra as well. So that's the first thing I'm going to look at here, and it's the mode of nutrition of bacteria, i.e. how do bacteria obtain their food? Exactly the same way that yeast obtained their food. So we're going to draw the very same diagram. So I'm going to start out with my dead matter. So the dead matter could be, for example, a piece of dead fish. That dead matter could also be a piece of raw meat. So we note then that bacteria can be spherical or rod-shaped. So I'm just going to draw a circular shape and I'm calling it a bacterium. And bacterium is just another word for one individual bacterial cell. So we note that bacteria are saprophytes. Now, saprophytes, we said before, they live off dead or decaying matter, but also they secrete enzymes into that dead matter. So saprophytes secrete enzymes into dead matter. We said these enzymes break down complex nutrients in the food to more soluble substances. And finally, these soluble nutrients are absorbed by the bacteria via their cell wall. So we'll just underline that. Soluble nutrients absorbed by bacteria via cell wall. So we'll just go back to our diagram again, and we show a diagram here for the enzymes being secreted into the actual food. So enzymes secreted. And then we'll take another one here then, how the soluble nutrients are absorbed. Now remember, you can use coloured pencils in your exam. A lot of the time students say, look, Sandra, not a hope, I had no time to use them. But you know, some people are able because they just write very quickly. And like I say, the diagram doesn't have to be very complex. As you're aware, my diagrams are always quite simple. And I do draw a lot, I write a lot, because the way I look at it is, there's no point in telling a student, learn that off or learn that diagram if I haven't drawn it myself. Because this is a problem sometimes where students are looking at a diagram so often. Oh yes, I know every part of that diagram, but can you actually draw it? It's really important that you sit down and you draw out these diagrams. So if you haven't been doing so up to now, maybe going forward, if we are drawing any diagrams, that you would draw them with me. And at least then you're making, using your time even more efficiently. So just to do a recap there, bacteria do not contain any chlorophyll. Therefore, in order to get a source of nutrition, they must get that food from an outside source. On our home economics course, we note that the bacteria that we talk about happen to be what are called saprophytes. Saprophytes secrete enzymes into the dead matter, whether it be dead fish, dead milk, chicken stock, milk, ice cream, soft cheese, all high in moisture. And these enzymes break down complex nutrients in the food. Then these soluble nutrients are absorbed by the bacteria via the cell wall, and we have a source of nutrition for the bacteria. So nice and simple. So that's our mode of nutrition. Now, secondly, we're going to look at our structure. Now, unless we have been living under a rock somewhere, I have said on many occasions that when we see the word structure on a home economics exam paper, whether it's structure of a refrigerator, or a refrigeration appliance, whether it's the structure of a yeast cell, the structure of meat, the structure of a wheat grain, the structure of yeast, the structure of bacteria, I could go on. It is nearly as common a term as classification. So structure, you draw your diagram and you give a written account. So you must give both. The only exception to the rule is if the question was phrased, draw and label a particular diagram. Otherwise, if you see structure and there are diagrams associated with it on our course, well, then you also give a written note. Now, I just want to remind you as well that when we were drawing the structure of meat or the structure of a wheat grain, I said to you, whatever parts you have labelled on your diagram, refer to those in your written note also. For example, I think 5 labels on the bacteria structure would be more than enough to know. So whatever 5 labels you give, make sure you refer to those very 5 terms in the written description. Now, I just want you to be aware, particularly those that don't do biology, not all bacteria have the same shape, which we'll cover in a few minutes. So when we say that we are learning the structure of a bacterial cell, also called a bacterium, I'm not saying that every bacteria looks like that, but the one that we have chosen and the one that's on our home economics syllabus does have quite a lot of similar features to the majority of bacteria. So this is just one example of a particular bacteria. In fact, looking at this, it could possibly be Salmonella. Now, how do I know it could be Salmonella? First of all, the shape, which is called rod-shaped bacteria. And rod-shaped bacteria, we'll look at in a few minutes, have straight sides and then have a curve at both ends. The other thing that makes me think it could be Salmonella, or Salmonella could look like this, is we have what are called flagellae. Now, flagellae are these, should we call them, now don't describe them as this in the exam, whiskers, or just see the way they're just like these little stringy bits, and they're at both ends. And what the flagellae do is that they help the bacteria to move, particularly in liquid. Not all bacteria have flagellae. Some bacteria have no flagellae. Now, on the home economics course, we call bacteria that have flagellae, we call them gram-negative bacteria. So this bacteria happens to be rod-shaped and gram-negative. Now, what do I mean by gram-negative? There was a scientist called Christian Gram once upon a time, and he developed this, I suppose, procedure and a way to using different stains to identify bacteria under the microscope. And as a result of this particular method, it was called after Christian Gram. If the bacteria have flagellae, they are gram-negative, and that means they do not produce what are called bacterial spores or endospores, which we'll be studying. And then bacteria under a microscope that have no flagellae, they are called gram-positive, which means they do produce bacterial spores, also called endospores. So the bacterium has also a jelly-like capsule. Now, not all bacteria have this jelly capsule, but certainly those that do, its purpose or its function is to protect the bacteria when they get to our stomach. In our stomach, we have what's called hydrochloric acid. And the majority of the time, the hydrochloric acid will kill bacteria. If the bacteria have the jelly-like capsule, they are protected from the hydrochloric acid, so they continue down the alimentary canal. Now, when we talk about probiotic foods that have live bacteria, majority of those bacteria will have a jelly capsule, because remember, we want to keep those bacteria alive right through our alimentary canal. We want them to arrive down in our colon. So that is our jelly-like capsule. Then we have fluid filling the center of the cell called cytoplasm. But before that, if we look at the dark black that we have here, this is the rigid cell wall. And just inside the rigid cell wall, we have a cell membrane. Now, if you wanted, you could put a broken line just on the inside for the cell membrane, or using a different color with a coloring pencil. So that would be our cell membrane. Very, very thin. I've exaggerated here, just inside the rigid cell wall. Then, like we said, we have liquid, which is the cytoplasm, and we call it granular cytoplasm, the same as we did for the yeast cell. And then we have what's called nuclear material present. The bacteria do not have a regular nucleus. We call it nuclear material. And finally then, we have what are called organelles, and organelles are suspended in the cytoplasm like the nuclear material, but two other organelles are ribosomes, and remember, we had food vacuoles, organelles suspended in the granular cytoplasm of yeast. So we have jelly-like capsule surrounding this particular bacterium to protect it from hydrochloric and the acid. Some bacteria are mobile and have these flagellae, so they can move around in liquid. And like I said there a few minutes ago, they are gram-negative. We have an outer rigid cell wall protecting the bacteria from pressure and keeping the shape. Inside we have our cell membrane, filled then the cell with liquid granular cytoplasm, and then we have organelles like ribosomes suspended in the cytoplasm, which are needed for protein synthesis, nuclear material called a nucleoid rather than a nucleus, which helps in cell development and growth of bacterial cell, and the nuclear material also contains enzymes and proteins. So we're just going to number there. 1, our jelly-like capsule. 2, we have our flagellae. 3, we have our outer rigid wall. 4, we have our cell membrane. 5, we have our cytoplasm. So maybe actually I'm going to change that. 6, we have our ribosomes, and 7, we have our nuclear material. So I think I'll actually change that, and we might as well just learn them all, just in case on a short question they asked us to label parts. So I'm just going to change that there to 7. So we're going to learn 7 labels which we have here. But just make sure that if you do have 7 labels on that diagram, like we have here, that you've 7. And I'm actually going to number them here as well. So I'm going to start with our jelly capsule. Then we have our flagellae 2. We have our rigid cell wall, which is 3. We have our cell membrane, which is 4. We have our cytoplasm, which is 5. We have our ribosomes, 6. And we have our nuclear material, which is 7. Now we do have an 8th one, which is the food vacuole, but you certainly wouldn't need all of them if you were drawing a diagram, but just to know that it stores food. and we'll just label our food vacuole then as 8. So that's our structure. Right, finally for today's lesson, we're moving on to the classification of bacteria according to shape. So the most common way, just apologies there, and to change that. And if you write the heading there, classification of bacteria. So classification of bacteria. Now, if I saw that on a short question, I'll show you how I would answer it in a few minutes. But the most common way of classifying bacteria is according to shape. So if we just move over here, so we'll take our heading, classify or classification of bacteria. So according to shape, we have 3 different shapes of bacteria, starting with what are called cocci. So number 1, cocci. Now cocci refer to spherical shaped bacteria, and cocci would be plural. So these are circular in shape under a microscope. Now, when you have 2 spherical shaped bacteria together under a microscope, this is called diplococci, di for 2. And anyone who would have pneumonia, the bacteria under a microscope causing the pneumonia would be diplococci. You've all heard of a strep throat, which is a very serious, it can potentially be serious, but it's, it can be very, very debilitating and extremely sore. So under a microscope, streptococci happen to look like a chain. So you've heard of strepsils, the throat lozenges. But if you had a strep throat and a swab was taken, the bacteria grown in a laboratory under a microscope, they would look like this, chains of spherical shaped bacteria. Then we have what are called staphylococci. Now, if you had something like a nasty boil or an infected spot and swabbed that, that would be staphylococci. And under a microscope, the bacteria would grow like a bunch, not as regular as this necessarily, but they would grow, or they would look like a bunch of grapes under the microscope. So skin infections would look like that. So it's our diplococci, staphylococci, streptococci, all spherical shaped bacteria, but they can arrange themselves in different ways. So this is our first way of classifying bacteria according to shape, cocci. Number 2, we have, like we had earlier, we have our rod-shaped bacteria. Now, rod-shaped bacteria look like we had earlier on, straight sides and then curved ends. Examples of rod-shaped bacteria on our course, Salmonella would be one. Any type of Clostridia. So Clostridia would be like Clostridium botulinum we look at in another lesson. And bacilli, any bacilli, remember we have Lactobacillus bulgaricus, Lactobacillus rhamnosus, Lactobacillus casei immunitas, we had Lactobacillus helveticus. So they have straight side, not like my dodgy crooked side there. So just to use a ruler in the exam. So straight sides and then our curve. And finally, according to shape, we have our third one, and these are the spiral shaped bacteria. Now, unlike cocci, which are definite spherical shape, or unlike the rods, which are a definite shape, spiral shapes can vary. And they can vary in terms of, you could have a comma shape right up to a shape that looks like that. Now, an STD, or sexually transmitted disease caused syphilis, SPHY, I can't even spell now. I'll have to go back to my notes. So would have that particular shape. Senior moment there. So 3 different shapes: cocci, spherical shaped bacteria, then we have rod shaped bacteria, and then we have spirals. And I would know an example for each. So starting with our spirals there, we have under a microscope, you've got curved shapes, anything from a comma to a coiled spring. And like I said, syphilis. If I can try and spell again, we'll go again, Sandra. It's a good thing we don't get docked marks for spelling, and that's a sexually transmitted disease, or STD. But under the microscope, it has a spiral shape. The cocci, then we have our diplococci, 2 together, streptococci, which are chains, and then our staphylococci in clusters like bunches of grapes. Then we have our rods, which have their definite shape, straight sides and curved at either end. And we said lactobacillus bacteria would be one there. And we gave examples of the cocci also. Now, that is the most common way of classifying bacteria: rods, spirals, and cocci. Now, on our home economics course, and I'm going to put NB here, please don't pass over this, because one year on a past exam paper, on a short question, they asked to classify bacteria according to their optimum temperature range. This is the range of temperature that they like best. So I'm going to divide up our group here, and we'll just highlight it actually all together, just so that it stands out. So we have what are called psychrophiles, mesophiles, and thermophiles. Now I'm going to just change that temperature range there to make it easier. Anywhere from 5 to 20, psychrophile. Anywhere from 20 to 45 would be a mesophile or a mesophile. And then sorry, that should be 75. No. And then 75 plus, we have a thermophile. Now, just want to go over here for a second. This appeared on a short question, so I'll just note here. And we have been looking at short questions in our online weekly grinds. So hopefully you understand what's going on here. So classify bacteria according to their temperature range. So as we know, short questions are worth 6 marks. There won't be 6 marks written beside it. So out with my pencil, and I'm going to write 2, 2, and 2. Remember, 2 marks, 2 subpoints. So they actually had psychrophiles written down, was in a box, mesophiles and thermophiles. So what you'd have to do then was you had to give the temperature range, but you would have said 5 to 20, then you'd have said 20 to 75, and then 75 plus, but always back up with an example. So a psychrophile could be listeria. So we're going to take listeria as an example. A mesophile would be salmonella. Note, I'm absolutely losing it here, that should be 45. Sincere apologies there. So because it has body temperature 20 to 45 salmonella, and in between is 37. And then thermophiles, then you have your Clostridium botulinum. So just to take a mental picture of that, 5 to 20, 20 to 45, and 75 plus. So I'll go back here, and I was correct the first time, so apologies about that. So as you are always, we can all make mistakes, and I'm certainly not perfect. And I'm going to put NB again. You know, often when I do things like this, I say if I've made a mistake in class and someone puts up their hand, which I would always encourage, you know, I said I will make mistakes and let me know. And then I'll say, this is an omen, are we going to see bacteria in our exam? So apologies about that. So just to make sure that you learn that well, because it's not on the biology course. Then we have another way of classifying bacteria, pathogenic and non-pathogenic. We already know from before, pathogenic bacteria cause disease, non-pathogenic do not. So pathogenic bacteria might cause TB, tuberculosis, diphtheria, brucellosis. Non-pathogenic are lactobacillus bacteria, which are found in milk. Now, then we have our fourth one, which is gram positive and gram negative. Now, to be honest, I don't see that they're going to focus too much on this, but I am going to put NB twice beside it. And the reason why I'm doing that is this classification, like the temperature range, is not covered in your or on your biology course. So we said about this scientist Christian Graham, he came up with this particular way of grouping bacteria using different stains, and then looking at the bacteria under a microscope. A reminder that if the bacteria have flagellae, they are gram negative. If the bacteria have no flagellae, they are gram positive. So what I'm going to do here at the side is draw a gram positive bacteria, no flagellae, and a gram, I'll actually use a different colour just to make it stand out a bit there, and a gram negative bacteria does have flagellae. So we'll just highlight key points that we need to note here. So gram positive produce spores. So positive, and they have a low resistance to antibiotics, example Clostridium botulinum. Gram negative do not produce spores, high resistance to antibiotic, and you could take salmonella. So I'm going to write in the center of each. So we'll just write here Clostridium botulinum. I'll just write C botulinum, and then I'm going to write salmonella here, just in case. It is on our course, so just to watch that. So what would I do then if I were asked on a short question to classify bacteria? Well, you're always going to try and push the boat out because of the fact that you're pitching for the high end. So possibly the marking scheme might only be according to shape, rods, cocci, and spirals. But this is how I would classify bacteria. I would classify, first of all, according to gram positive or gram negative. And give your 2 examples. Remember, you are allowed to do what I'm doing here, right like this. So gram positive, they have no flagellae, and they produce spores. Gram negative, so they're gram positive, and then gram negative have flagellae, and they do not produce spores. Then our second way, you could say pathogenic, disease causing. So you could talk about Clostridium botulinum. Or non-pathogenic, like lactobacillus and bacteria. And then thirdly, you might write shape. And then I would put a little asterisk, and I would have most common. And then I would have rods, cocci, and spirals. Rods would go with salmonella, cocci, bacteria that cause pneumonia, or even better still, we could have our streptococci. And then spirals then, which can cause a word I cannot spell. And that would be syphilis. Now, some people are going to look at that and say, I've lost my mind. The way I'm looking at that is, I want the perfect paper. I want to show the examiner that I absolutely know my work and I deserve 1.5% for that answer. And I'm also demonstrating that I have a knowledge beyond what the average student has, where they might just talk about the shape. And I also know that I have 2 full minutes to write that. And I didn't take 2 minutes to do that. But that's what my students have been using or doing for the last 30 years, pushing the standard all of the time. And that completes today's lesson.